Jozef from Belgium. I'm sorry for the brief introduction you had about my country earlier, but I did my PhD back in Brussels working on cystic fibrosis infections. Then I flew all across the ocean to work here as a postdoc researcher at BU studying biofilms. So I'm going to talk about how to interrupt a bacterial party, also known as a biofilm. And since the topic of these research days is rethinking research, I would like to start with some misconceptions that most people have when they think about bacteria. First of all, most people think that all bacteria are actually bad. And second, most people see bacteria as microscopic creatures that are always unicellular. In the next few slides, I will prove you that this is actually wrong. So, the majority of bacteria are actually good, such as the bacteria living in our digestive tract that help us digest food and produce vitamins, but also the bacteria that are associated with plant roots that actually promote plant growth. Unfortunately, there are some bad bacteria. Then they can actually mess up our gut microbiomes, causing diarrhea, or you see some nasty pictures here of skin infections and eye infections that are caused by bacteria. Furthermore, they can also infect plants. So bacteria are actually very social, so they can live as single cells, as you see on the left, but they can also live closely together in structures called biofilms. And those biofilms are actually antibiotic resistant, which is a major problem, as you will see in the next few slides. So this is how the biofilm life cycle happens. First, single cells will attach to a surface, they will form smaller clusters, that eventually will develop into mature biofilms that have the antibiotic-resistant characteristics. So here you see an example of biofilm formation by Xylella fastidiosa, which is a plant pathogen. And you see that single cells quickly will cluster together and form macroscopic structures that you can just see with your naked eyes. Another example is Pseudomonas aeruginosa, which is a nasty opportunistic pathogen that can cause cystic fibrosis. So initially, single cells are dividing on a surface, and that will actually keep on dividing until the entire surface is covered with biofilms. So biofilms can also be seen in nature. You can see them in Yellowstone National Park, for instance, but closer to home, you can also see stromatolites formed by biofilms of cyanobacteria in Green Lake State Park, New York. However, biofilms can also be very nasty. So in cystic fibrosis patients, on the upper left, you see Pseudomonas aeruginosa biofilms, but biofilms can also be seen on teeth, causing dental plaque formation, or they can slow down ships by attaching to the hulls or clogging up the drain pipes. So why are biofilms so bad? Well, biofilms protect the bacteria from the immune system as well as antibiotics. And this protection from antibiotics leads to antimicrobial resistance, which is a major problem for the next few years and the next few decades even. It is estimated that by 2050, about 10 million people will die because of antimicrobial resistance, which is even more than the 8.2 million deaths we have right now from cancer. So this is a major problem, and in the Sauer lab, where I'm working, we are actually trying to tackle this problem by studying biofilm development. And we are trying to look at pathways that lead to the interference of antimicrobial resistance. So we try to reverse the antimicrobial resistant problem. So how can we stop biofilm formation? We can follow several strategies. One of them is reversing the biofilm formation stages or late stages. Or we can interfere at the initial stages of biofilm formation where tolerance to antibiotics is actually activated. I'm working on this one particular protein called SAGS that actually acts as a toggle switch, allowing the bacteria to transition from the planktonic to the biofilm stage of growth. And we are looking for mechanisms that turn this switch off, making the bacteria treatable by antibiotics. So this protein SAGS actually acts as a sensor that catches a signal from the environment and then will initiate a mechanism that leads to biofilm development, but also antibiotic tolerance. And recently, we actually identified two key residues in the sensory domain of this protein that are involved in antibiotic tolerance or biofilm formation. And by blocking those residues, we could actually reduce the bacterial loads in laboratory mice, which were a lot happier after this happened. So I would like to summarize my talk by giving you some take-home messages. One of them is that not all bacteria are harmful, and the bacteria are actually very social, and they can grow as biofilms that protect them from antibiotics. However, we found one molecule that we can target to prevent antibiotic resistance, which is great for future applications. I would like to thank all the people I've worked with so far in the past 3.5 years, and I would like to thank you for your attention.